Fungal virulence and pathogenesis. The purpose of this presentation is to enable students to be able to recall what has been discussed during the classroom lectures so that they can play the video several times in order to grab the specific ideas discussed during the normal class lectures. Now, virulence factors of fungi. Generally, these are important characteristics that determine the ability of pathogenic fungi to cause diseases. They are generally important processes or mechanisms used by fungi to establish a disease. It involves certain adaptive features of the fungi as well as possession of particular virulence factors, as we are going to see. Now, the first important virulent factor used by fungi is the adhesion. Adhesion is defined as the ability of the pathogen to attach themselves to the specific receptors on the host cells. In the case of fungi, fungi attach themselves to specific receptors on the host cells using three major ligands or projections on the surface of the fungal cells, which are generally called adhesins. Now, these adhesins, as I said, are surface projections that are involved in specific attachment of fungal cells to specific receptors at the portal of entry of the host. Usually, they interact with and bind to the receptors of the host in specific fashion, which we can say is comparable to that of enzyme and substrate interaction. Now, the first of these three ligands are the agglutinin-like sequences. These agglutinin-like sequences are a group of 7 glycosylated proteins, agglutinin-like sequence 1 to agglutinin-like sequence 7, which are involved in the fungal attachment to the host tissues. They specifically adhere with the human buccal epithelial cells. They adhere with the human buccal epithelial cells and fibronectin tissues, collagen in the muscle tissues, laminin, as well as endothelial cells. Now, another important ligand is the fungal hyperwall proteins, which are a group of proteins that also aid the fungi to attach to the host cell. Now, these fungal hyperwall proteins they specifically bind with important cells such as the human buccal epithelial cells. The next category is the integrin-like proteins, of which integrin-like protein 1 is the most important one involved in fungal pathogenesis. This also help fungi to attach themselves to the receptors of the host cells, especially Candida species. But the binding to the host cells in the case of integrin-like proteins is also is enhanced by the presence of tissues such as fibronectin, laminin, and as well as collagen 1 and collagen 4 on the host cells. So these are the ligands involved in the adhesion or attachment of the fungal cells to the host tissues. The next category of virulent factor, which is attached to the adhesion is the biofilm formation. Usually after initial adhesion, the next stage is formation of biofilm by the fungi. For biofilm formation is generally the ability of the fungi to systematically arrange themselves in masses or aggregates to occupy a particular portion of the host body, so much so that they cannot easily be washed away. Now, this biofilm formation takes place in three main stages. The first one is the adherence. Adherence involves irreversible attachment of the fungal cells or tissues using so many adhesion factors such as the hyperwall protein we talked about, the integrin-like proteins, agglutinin-like sequences, and so on. This stage in fungi is controlled by certain genes such as the SUN3 gene, NUP85, MDS3, and KEM1 genes. Now, immediately after adherence, the next step is the growth. The fungi will now begin to grow after being able to adhere to the host. The next step is growth. So the growth of the fungal cells begin through hyperelongation and production of exopolysaccharides, which makes the cells firmly stick to one another. This particular stage is enhanced by production of farnesyl protein, which prevent filament formation and enhance hyperelongation. So in the presence of many polysaccharide produced, the cells become firmly attached to each other. And another important factor involved in the growth is aspartyl proteinases, ASP enzymes, which enhance more nutrient utilization and adherence by the fungi to the host cells. Usually, these proteinases break down proteins in the host cells to enable more nutrient, to make more nutrient available to the fungi. So the fungal cell will utilize the proteins to multiply. Now this stage is also controlled by a hyperregulatory gene, which is called EFG1 gene. Now, immediately after growth, the last stage in biofilm formation is maturation of the biofilm. Now, the maturation stage is involve hyperelongation to form a mass of mycelium. So the hyphae will elongate by several cell divisions to form a mass of mycelium with altered phenotype, which will adhere more to the surface of the tissues. In this stage, generally, more exopolysaccharides are produced, and this particular stage is controlled by tyrosol. Tyrosol acts antagonistically to farnesyl, by inducing filamentation, which was restricted by farnesol. So it induced filamentation, so more filament will be produced at this particular stage. And once this stage is reached, the fungi cannot easily be washed from the portal of entry. That is to say, it has established itself firmly to the host cells. Now, the next virulent factor of fungi after biofilm formation, another important virulent factor is morphogenesis, which is otherwise called dimorphism. Dimorphism is just a transition from unicellular yeast form to filamentous form and vice versa. So it involves the transmission either from mold fungal form to yeast form. This particular virulent factor usually is utilized by fungi to spread from the portal of entry. Once they establish the biofilm, then dimorphism can occur, in which the fungi will spread via the bloodstream in form of unicellular fungi to the target site where they can establish a diseases. So the transition from yeast to mold form is facilitated by the presence of optimum growth conditions, such as the presence of nutrient, pH, temperature, and so on. Whereas the transition from mold to yeast is mostly an adaptive mechanism developed by the pathogenic fungi against the harsh environmental conditions of the host, such as the temperature, change in temperature, pH, and nutrient, etc. This morphogenesis, as I said, is very important in fungal pathogenesis because dissemination of pathogenic fungi is mostly achieved in yeast form than in mold form. However, the mold form is utilized when causing damage to the tissues and evading host defenses. So, on reaching the target organ, the fungi can also change from yeast to mold form, where it can cause damage to the host tissues and prevent phagocytosis. Now, this morphogenesis is generally controlled by certain genetic factors, such as the PHR1 gene, EC1, HYR gene, and so on. Apart from this morphogenesis, the next virulent factor, which is very important also, is phenotypic switching. Phenotypic switching involves change in appearance, biochemical characteristics, or metabolism of the fungi. This switching is highly advantageous to many pathogenic fungi because it makes them to recognize certain barriers in the environment and rapidly develop a means of adaptation to such changes by expression of various metabolic pathways, enzymes, etc. The phenotypic switching is reversible and can occur spontaneously during stress condition. In addition, the switching may also make a less virulent fungi to become more virulent type. Now, the next virulent factor is production of phospholipase enzyme. Phospholipase enzymes are enzymes that hydrolyze ester bonds, which are in form of glycophospholipids, thereby breaking the barriers in the host cell plasma membrane and facilitating the fungal invasiveness. Generally, there are 4 groups of phospholipases: phospholipase A, phospholipases B, C, and and D, of which phospholipases, subdivisions, they have subdivisions, of which the phospholipases B1 plays a major role in the cleavage of phosphodiester bonds. In the phosphodiester bonds in the host cells. Now, usually this phospholipases is found at the hyphal tip, the tip of the hyphal cells usually produce this important enzyme which helps the hyphae to penetrate into the host cells by breaking down the phosphodiester bonds. Now, another group of enzymes that also helps the fungi to break down host cells and separate are the proteinases enzymes. These proteinases enzymes break down proteins in tissues, thereby facilitating colonization of the fungal pathogens. Tissues generally, as we know, they serve as barrier to pathogenic fungi during adhesion, colonization, and invasion due to their complex proteinous nature. Therefore, fungi that have proteinases have more ability to cause infection compared to fungi that do not produce proteinases enzyme. So this proteinases enzyme is a very, very important factor that is utilized in disease causation. Some of the complex proteinases enzymes utilized by fungi are the aspartyl proteinases. These aspartyl proteinases hydrolyze proteins such as albumin, hemoglobin, immunoglobulins, keratin on the skin, collagen in muscles, and so on. There are about 10 different types of this ASP that are used for degradation of complex proteins. Generally, this ASP carry out the hydrolysis of several proteins if we want to list their importance. One is that they hydrolyze several proteins, releasing many nutrients from the host cells, which are in turn used by the fungi to survive and establish infection. Number two, they also cleave peptide bonds of membrane proteins and mediate morphogenesis of the fungi from yeast to more invasive hyphal form, leading to rapid invasion and tissue damage. Number three, they mediate adherence of the fungi to host tissues by damaging the tissues and exposing them to the fungi, leading to increased expression of other adhesion factors such as agglutinin-like sequence and so on, resulting in more strong binding and subsequently biofilm formation. Now, another important virulence factor which we need to talk about is the thermotolerance. This is the ability of the fungi to adjust to survive and replicate within the range of the body temperatures of the host. Fungi mostly grow under low temperature, and hence any pathogenic fungi that can develop a means of tolerating the high temperature of the bodies will have more advantage of causing diseases. So in general, this thermotolerance is the ability of the fungi to adjust themselves to tolerate the high temperatures in the host tissues. Usually we grow, if we want to grow fungi, we grow them at a temperature of room temperature 25 degrees Celsius, but the temperature of the host is usually 37 degrees Celsius. So pathogenic fungi have this ability of thermoadaptation by producing a kind of the heat shock proteins, which gives them the ability to withstand the high temperature of the host. It also assists in dimorphism, especially from mycelial form to the yeasty form transformation, just to be able to survive in the body temperature of the host. We also have the cell wall component and capsules. Cell wall component and capsule in certain fungi also increase the fungal virulence because they protect the fungi against host defenses. They generally prevent phagocytic destructions by the host cells. Example, the alpha glucan, beta glucan, glucoxymanan, all these ones helps the fungi to evade phagocytosis. They help the fungi to resist phagocytosis by the host cells. Especially, they also, in addition to that, glucoxymanan, for example, helps the fungi to cause diseases such as pneumonia. Fungi that cause pneumonia usually utilize glucoxymanan, leading to the formation of the symptoms such as meningitis and pneumonia. A good example of this fungi are Cryptococcus neoformans and the likes. Okay, now having discussed the virulent factors used in disease causation, now we'll discuss another important virulent factor. The previous virulent factors we discussed are utilized by fungi to actively cause diseases in the host. That means the fungi has to be in the body of the host in order to establish the diseases. Now we have a situation in which the fungi secrete toxins. That is, they cause diseases by means of toxogenicity. Toxogenicity is the production of toxins by fungi, and these toxins are called mycotoxins. Toxins produced by fungi are called mycotoxins, and the diseases as a result of mycotoxins are called mycotoxicosis. Now, mycotoxins, when produced by fungi, they produce them on food products, and once the food substances are consumed, they establish a disease condition known as mycotoxicosis. So these mycotoxins are low molecular weight toxic metabolites that are produced by pathogenic fungi, and they have the ability to elicit toxic response in humans and animals. They are mostly associated with contaminated farm products, food, and other products that are produced from the farm, as well as stored products. So they are associated with several outbreaks of foodborne intoxications. They are very potent in such a way that very small amount of mycotoxin can cause a death of large number of animals. And in addition, most mycotoxins are highly carcinogenic. Apart from ability to cause food poisoning, death, and so on, most of them are highly carcinogenic. The four common mycotoxins that are usually involved in disease causation include aflatoxin, trichoticins, zearalenone, and pulmonicins. These four are the most common mycotoxins associated with mycotoxicosis. The first one, as we said, is aflatoxins. Aflatoxins are generally fungal toxins that are isolated from Aspergillus flavus originally, but later it was found to be produced by several species from the genus Aspergillus. And that is why the toxin is named after the fungi, Aspergillus flavus toxin. Most common members that produce these toxins are Aspergillus parasiticus and Aspergillus flavus. They produce a variety of these aflatoxins, which are about 20 different types. However, the most important ones that are involved in disease causation are aflatoxin B1, aflatoxin B2, aflatoxin G1, aflatoxin G2, aflatoxin M1, and aflatoxin M2. Now, as we said, these fungi contaminate food in the field and in stores, leading to a type of mycotoxicosis, which is called aflatoxicosis. This aflatoxicosis can be acute aflatoxicosis, which is a very short-term aflatoxicosis characterized by consumption of large amount of aflatoxin in food, leading to cases of diarrhea and vomiting, hemorrhage, systemic toxicity such as liver toxicity, kidney toxicity, brain damage, and so many other systemic diseases which can lead to instant death. It can also be disease due to aflatoxin can also be chronic aflatoxicosis. Chronic aflatoxicosis is the most common now in our society, which is characterized by consumption of very small doses of aflatoxin over a long period of time. These doses of aflatoxins accumulate in the body tissues, leading to several diseases. Most important one is cancer. And the most common is hepatocellular carcinoma, which is liver cancer. They also cause cancer of the respiratory tract cancer. They also cause sterility when they affect the reproductive system. They also cause malnutrition in children when their food contains smaller doses of these aflatoxins. They lead to malnutrition. In animals, aflatoxin cause several diseases in which you will see a complete poultry farm being a complete poultry farm being involved in the outbreak. So you'll see all the chickens being slaughtered, or most of them die as a result of the toxic effect. Now, another important mycotoxin is the trichoticins mycotoxins, which are produced by Fusarium trichodioides and Fusarium graminearum. There are about 150 trichotoxins, mycotoxins that are produced. The most important one among them is the trichotoxin 2 toxin, which causes trichotoxicity. An example of this toxicity is the alimentary toxic aleukia. Alimentary toxic aleukia usually is characterized by burning sensation in the mouth, in the tongue, in the throat, esophagus, and stomach, the whole gastrointestinal tract, and severe gastrointestinal disturbances, especially in cattle and other animals. Now, another mycotoxin is zearalenone. Zearalenone is a form of phenolic salicylic acid lactone toxin, which is also produced by members of Fusarium species, such as Fusarium trichodioides and Fusarium graminearum. This particular toxin affects mammals, leading to hyperestrogenism, swollen vulva, and mammae. So is most common in farm animals that feed on contaminated zearalenone contaminated animal feed. Now we have the last one among the 4 important mycotoxins, which are the fumonisins. Now, fumonisins are also a group of mycotoxins that have several classes. However, the most important one is the fumonisin B1, which is produced by members of Fusarium, also Fusarium species. And this particular toxin has been found to cause toxicity and mortality in horses. It causes animal diseases called the equine leukoencephalitis, characterized by inflammation of the brain and then death of the affected animal. So these are the 4 major group of mycotoxins that are produced by fungi, which have the ability to establish diseases. Now, having said that, let us look at the common pathway in which mycotoxins are transmitted along the food chain to reach human beings. This one is a pathway of aflatoxin transmission along the food chain. If we look at it, it all started from the contaminated foods. The foods contaminated are either utilized to make animal feed, such as livestock feed. And if they are used to make livestock feeds, they end up being consumed by the home animals, cows, cattle, sheep, and so on. So when these animals consume aflatoxin contaminated livestock feed, the products of these animals become contaminated with the mycotoxins, such as the meat, the milk, the eggs, all become contaminated because aflatoxins, just like other mycotoxins, remain persistent along the food chain. It is destroyed, it becomes denatured at a temperature of 270 degrees Celsius, which is far above the cooking temperature. That means the toxin can be transmitted in the food, and then even after cooking, it can remain persistent until it gets to human beings. So generally, after affecting the animals, the animal product, the products from those animals end up consumed by humans, and when humans consume such products, they develop either acute or chronic aflatoxicosis, as we said. Another way is that some of these farm products are directly consumed by human beings. The other way we discuss is just like an indirect way, but this one is is a, is a direct way of aflatoxin transmission, in which it affects the humans directly. Now, whichever way, aflatoxins causes either chronic or acute toxicity once consumed by the humans. Now, another important thing we want to discuss is the general mechanism of fungal pathogenicity. Pathogenicity is the ability of a pathogen to invade host tissues and cause diseases with specific signs and symptoms. So, on the other hand, we have pathogenesis, which refers to stepwise processes followed by the pathogen to cause diseases. So, pathogenesis is a stepwise process, while pathogenicity is the inherent ability of the organism to cause diseases. In fungi, the process of pathogenesis take place in 5 major stages. The first stage in the pathogenesis is the adherence. As we said, for a fungi to cause diseases, they utilize adherence to the host tissues. So this adherence, as we explained earlier, involves participation of two factors: the specific receptors on the surface of the host cells and tissues, which are the carbohydrate and peptides, and then the ligands or adhesins on the fungal cells. So both of them interact just like enzyme and substrate. So they interact in a specific fashion. And as we mentioned earlier, so many of these ligands are produced by fungi, such as agglutinin-like sequences, fungal hyperwall proteins, integrin-like proteins, and so on, which are used by the fungi to ensure attachment to the host cell. So once the attachment is established, the next thing is entry. The fungi will now enter the body, taking the advantage of the common portal of entry, natural openings in the body, such as the gastrointestinal tract, gastrourinary tract, respiratory tract, the eyes, the ears, broken skin, and so on. So once the fungi attach, it will now enter, and immediately after entry, the next step is for the fungi to adopt. So this stage is called adaptation and propagation. It involves the survival of the fungi after entry into the body, and it is generally achieved through expression of many virulence factors such as morphogenesis, phenotypic switching, thermotolerance, as we discussed. In mammals, there may be change in the mode of nutrition from the saprophytic mode of the fungi to parasitic mode. So based on this, the fungi will now propagate. It will now begin to it will multiply in numbers, and the next step is dissemination. Once propagation is achieved, that means the fungi has already adopted to the environment of the body and has multiplied, the next thing is dissemination. It will now spread by trying to evade the host defenses in the body. It will now spread to barriers to reach the target site where they can establish a diseases. So they will spread using the virulence factors as we mentioned earlier. Now, after dissemination, the next step is damage to the host cells. Once the fungi disseminate to reach the target organ, it will now begin to cause damage or diseases in the affected area. And this is generally achieved after colonization and dissemination to particular locus in the body. The fungi exert damage to the tissues through enzyme secretion, hyperpenetration. Some of the tissues that are commonly damaged by fungi include the meninges, membranes of the brain and spinal cord, the lungs, the skin, vagina, vocal cavity, blood, etc. So this one, generally the damage is achieved through the extracellular enzymes and hyperpenetration. So that will lead to disease causation in the fungi. Now, for any agent that cause diseases in the body, host defenses usually try to prevent the body against such particular agent. In the case of fungi, we have the host defenses that also act against fungal infection. So host defenses against fungal infection generally involve the non-specific or innate immunity. Non-specific or the innate immunity are the most involved in fungal infection, because most of the fungal infections are the skin infection, the surface layer of the skin, the dermal layer, the subcutaneous tissues, where most of the defenses around those areas are the primary defenses, except for some fungi that causes systemic infection. That is where adaptive immunity comes into play. So this innate defenses against fungal infection is divided into three forms. We have the cells and tissues that are involved, that are part of the innate defenses. These cells and tissues usually phagocytose and kill fungal cells. They include the granulocytes, the macrophages, which are the phagocytes, the dendritic cells, the natural killer cells. All these cells attacks and destroy fungal pathogen. Another important natural barrier to fungal infection are the physical barriers. These physical barriers include the skin and mucous membrane. These barriers are very tough. They contain the keratin and other secretions that serve to prevent a lot of fungi from causing infection, except for those highly pathogenic fungi that possess keratinases enzymes that break down the keratin and so on to establish diseases. So these barriers helps to prevent infection against many fungal pathogens. Then we have also the chemical mediators. Chemical mediators also act in a non-specific fashion, just like the physical barriers. They include most of the enzymes and proteins, like the defensins, lysozyme enzymes, which we all know very popular, that lyses the cells of pathogenic organisms. And then we also have the complement system. These complement systems are system of serum proteins that are capable of eliciting various biological effects in the immune system, leading to lysis of the target pathogen. They opsonize, they identify a pathogen, opsonize the pathogen, and then they lead to several cascade effects, which finally lead to the destruction of the pathogen. In essence, these are the little I will provide for this particular topic. I hope the student will utilize this video effectively to help them recall what has been discussed during the general classroom lectures. I thank you all and I wish you best of luck.